In the next stage, that's going to enter our first talk of today from our honorary guest speakers. Honorary guest speakers, Dr. Professor Jeremy Nicholson และในลำดับต่อไปค่ะเราจะเข้าสู่ทอล์กแรกปาฐกถากิตติมศักดิ์ในวันนี้ค่ะในช่วงแรกนะคะเราได้รับเกียรติจาก Professor Jeremy Nicholson เป็นวิทยากรท่านแรกวิทยากรพิเศษของเราในวันนี้ค่ะ so after this is going to be English only และจากนี้ไปค่ะเราจะดำเนินการเป็นภาษาอังกฤษเท่านั้นนะคะ so let me introduce our first guest speakers for today our honorary speakers Professor Jeremy Nicholson. So he is an esteemed academic and is well known as one of the pioneers of biological NMR spectroscopies, metabolic profiling, and systems medicines. His major research focus is on the development of diagnostic and prognostics and molecular phenotyping analytical technologies, and to apply such innovations to personalized healthcare, to metabolic disease, and populations phenotypings. He obtained his PhD from King's College London and has held various academic appointments and leadership positions. He is currently an emeritus professor at the Imperial College London, Pro-Vice Chancellor for Health Science at Murdoch University, Australia, and he is the director of Australian National Phenome Centres. Throughout his career, he has received numerous research prizes and has been recognized as the highly cited research in pharmacology and toxicologies. He holds visiting honorary professorships at several universities, including Fudan Universities, Nanyang Universities, the Mayo Clinics, and many, many more. He is also the Fellows of the UK Academy of Medical Science, and he has received honorary lifetime memberships from various societies and institutions. It has been the greatest honor to have him with us today. Please join me in welcoming Professor Jeremy Nicholson. Please give our big hand of applause. Thank you very much.
Okay, I might just sit down. That's interesting. Do I have somebody provide me with a laser pointer, please? Obviously, I'm considered too old to stand up. Okay, that's good. Well, it's a great pleasure to be here today, ladies and gentlemen, to talk to you about a subject that's close to my heart, which is molecular phenomics. Hang on, is that the pointer? Where's the pointer on that? Pointer. No, that's a slide advancer. You've got a pointer, a laser pointer. No, I don't want that. All right, it's all right, don't worry. Yep, it works now. Interesting. Okay, right, after that brief pause. So what I'm going to do today is to give you a sort of historical perspective on molecular phenomics and what it involves and why we think it's important. You can think of the 20th century as being the century of the genome. The 21st century is going to be the century of the phenome, where we look at and try to understand the interactions of genes with the environment in order to understand where diseases actually come from in the human population, and to try using these appropriate technologies to stratify people, individuals, populations into disease risk groups, and also when they get diseases to stratify individual disease groups into subsections so that we can understand better how to monitor their therapy and intervene for their benefit. So I've been asked to say about what a phenome centre is. So this is a snapshot of our phenome centre in Australia. And it's a very large, well, somebody needs to turn that down. Well-equipped laboratory using very high-end spectroscopic and chemical tools, which can provide a basically multivariate description of the biochemicals in your body, which are related to your likelihood of getting a disease. So our lab is situated on a hospital campus, so we're very close to patients, but it also has much broader capabilities, which I'll explain. So what does the ANPC actually do? Well, it's looking at dynamic healthcare challenges that are relevant across the world, and in particular with angles related to personalised and public healthcare. And part of understanding the changing world is understanding the complexity of the world and how things, biological features of the body interact with themselves and also the environment. There's a lot of technology that goes into developing a phenomic program and a phenome center. I'll tell you a little bit about the analytical technologies involved. There's also a huge amount of informatics and mathematical processing of data to bring it all together and try to produce what we call systems models to describe the disease processes. And then ultimately, the data and the models that we develop, we want to use for real people. So there's a job of clinical translation, which is moving it from the laboratory, from the model into the hospital environment and the patient, and to enable what we call clinical actionability, so that a doctor can look at a piece of data and go, I know what to do next with that patient. So in Australia, we have multiple modalities of technology, but also how they interact with our various research programs. So we're interested in human population phenotyping, trying to model thousands and thousands of people, trying to look at future disease risks. We're interested in individual patients using metabolic and other profiles to characterize the disease state and stratify them. But a lot of the technology that we use is relevant to other parts of science as well. So it's very relevant to understanding what foods do in the body, nutrition, and also the molecular composition of food, which is obviously relevant to the health properties of those foods. And foods are made in the environment by people who do agriculture. So the technologies are also relevant to refining and optimizing agricultural technologies. So in Australia, we have all of those coming together in one laboratory. And our future trajectory is probably to develop a series of other laboratories that act as spokes to the central hub. So there'll be a laboratory of environmental phenomics, a laboratory of veterinary phenomics, a laboratory of agricultural phenomics, but that's for the future. So we're all shaped by our gene-environment interactions. We have these genes that we're born with, and then all the different things that happen to you during your life, the things you eat, how you exercise, your lifestyle, all interact to create your individual phenome, the product of the gene-environment interaction. But all those things also interact to create your future disease risks. So for most people, 90 plus, 90, maybe even 95% of people in the world ultimately die from adverse environment to gene interactions. Most of the way we die is actually not related to our genes per se. And of course, all these things interact dynamically throughout life to create this evolving phenotype. And the expression of the outcome of gene environment interactions is called the phenome. And phenomics is the measurement of all of those different properties. A phenome is an emergent property set, and it's represented by a whole series of variables. It can be physical variables, your height, your weight, things like that, eye color, all of those things, some of which are genetically determined, some of which are environmentally determined, but also all the chemicals that you can measure in the body. And there's hundreds and hundreds of thousands of those, the whole range of molecular weights going from big things right down to little tiny molecules. And all of that is expressed in the phenome. Now, we have a very dynamic world where the healthcare challenges are changing all the time. And our aspiration is to be able to take biological knowledge, biological data that we measure from individuals and to use those data to optimize interventions and therapy. So personalized or precision medicine is about the biology, measuring the precision biology of the patient, and based on that knowledge to intervene more effectively. And precision medicine is a very complex process. There's lots of people have different ideas of what precision medicine is. A lot of people think it's all about genetics, measuring genomic profiles. That's important, but it's only a small part of the story. There's also all your metabolic patterns, and of course, the other sets of genes that live within you, the microbial genes, the microbiome, etc. And all of that knowledge has to be put together and modeled in a way that is approvable by society. And that means there's interactions with the key stakeholders in precision medicine, the pharmaceutical companies, the educators, the regulators, etc. So it's a very complex process. Why do we need precision medicine? So why are we doing it? It's to get better, of course. Diabetes happens to be a particularly good example of where we need precision medicine. So if you look at this little diagram here, this has got sort of all the different drugs that people use in the order they use them in relation to early onset diabetes. So for instance, this thing here is metformin. So if somebody's suspected of having diabetes, the first thing they're going to try and correct it with is probably metformin for most cases. But there's actually something like 16 different interventions at the moment clinically available for diabetes. So which is the right one for the right person? Because after you've done it, metformin doesn't work, and it becomes a bit of a lottery. And I introduced the sort of idea of diabetes roulette, where basically the doctor goes, well, that hasn't worked, let's try this one. And whatever they choose is based on their experience with other patients, but it's not based on evidence. And we, in precision medicine, we want this to be evidence-based and based on real science. And so diabetes is a good example. The other thing that we want in society is to live for longer and to be healthier. So the 100-100 concept, it's been around for a number of years now, is the idea of living 100 years of healthy life. And living to 100, well, it used to be quite, it's got easier as medicine has got better. But those last 20 or 30 years are actually more difficult. So we can live 100 years, but maybe the last 20 or 30 are not so good because you're not so healthy. So we have to think about how we monitor that and improve lifestyle management. There's been a huge step backwards very recently due to COVID-19. And as a result of the combined effects of COVID-19, not only acutely, but also what we call, the excess mortality due to the way we've changed using hospitals. In fact, life expectancy now is lower than it's been since 1996. So all of that fantastic science that's gone into improving human healthcare over the last 30 years has been reset in a very short time period by COVID. So that is quite extraordinary. And so in the United States, that amounts to 3 years of life expectancy drop in the United States. That is truly scary. And people should be screaming in the streets about this, but they're not yet, probably because they don't really know it or understand it. So we have to think about what kills us now and then. And we've changed the patterns of mortality over the years through the use of science and medicine, and in particular through the public health and sanitation, which made massive effects in the 19th and late 19th and 20th century, but also the use of antibiotics. And if we look what's changed, we've gone from basically people dying of infectious diseases, to people dying of diseases that we think of being related to old age. So cardiovascular disease, dementias, and cancers and things like that. So we've shifted the balance. And if we look at the leading causes of death worldwide, cardiovascular and cardiometabolic diseases are right up there. There's cancers, of course, there's respiratory diseases, etc. But if you think about what COVID's just done, it's changed the probabilities of adverse outcomes for vast numbers of the population. When I say vast numbers, I mean hundreds of millions of people around the world now have inferior cardiovascular risk profiles and also more likely to catch diabetes and also have dementias as well. So COVID, people think of it as an acute respiratory disease, and it is, but it has so many other knock-on effects that are impacting on global health problems for the future, and massively impacting on the cost of this. So the costs of COVID in the 21st century will be in the trillions of dollars, right? And most of that cost is still to come because of the changes in future healthcare patterns. We were our own worst enemies. So, you know, in America, people sit around, watch themselves get fat. That's something the pattern of obesity change in the States. So, United States over about 25 years, so slightly out of date now. The whole of the map is red. About 35% of Americans are clinically obese. And that has a whole series of other knock-on effects as well. Around the world, lifestyles are changing. There's a westernization of China. So China now has got McDonald's and Starbucks and all the other things. If you westernize, you tend to get Western diseases, because a lot of Western diseases of modern times are driven by lifestyle and eating behavior. And if you look at this in China, for instance, we now know that type 2 diabetes is driven by BMI, body mass index, how fat you are, basically. And these circles represent the total world burdens of these type 2 diabetes. The big orange circle is China, and that one is India, and that's the United States. But what is instructive is to think about how that is driven in a relatively short time by changes in BMI. Look at the way that country burden of diabetes increased over just 24, 25 years in China. It's quite scary. So what that tells you immediately is type 2 diabetes is not driven by genes, right? It's driven by really by environment and dietary lifestyle, because can't be driven by genes, because that's less than, you know, a human lifespan by a long way. It's making things worse because there isn't enough insulin in the world, and it's estimated by 2030 about 40 million people in the world will not have enough insulin, simply because there are finite resources available for producing insulin at scale. So if you make more diabetics, you can't necessarily make more insulin. So that's really seriously worrying. And also, if you look at COVID, that has had a massive increase on the number of diabetics around the world, and we have a major research program on that. I will talk to you about that tomorrow. Also, these things are often set in early life. So if you look at early antibiotic exposure, it actually has influences on the development of obesity, adiposity as they call it, which we know drives diabetes. So the chance of you being a fat teenager is set actually by the time you're 2 years old. And this is a study a few years ago, which from a Scandinavian group, that showed the microbiome, the microbes inside you, at the age of 2, accurately predict the obesity when you're about 13 or 14. So it's set very, very early. And in fact, many childhood diseases are set early as well. And our children are our future. So if you look in the UK, half of the children in the UK don't exercise enough. About 30% of them drink alcohol, and 10%, we're talking about teenagers now, actually smoke as well. And put that together, those are all drive inflammation. And inflammation underpins almost all major non-infectious diseases, and is the result of infectious diseases as well. So the risks are changed for all the things that kill us by things we do in childhood. One of the mediators of this is the microbiome. So there are microbes on every surface of our body, every epithelial surface, in our noses, in our lungs, in our gut, and they are the interface between environment and us. So in order for an environmental influence to get through to our cells and genes, it has to go through a layer of microbes. And unsurprisingly, there are lots of disorders of the microbiome now that have been recognized that are associated with a huge range of different types of disease, which you wouldn't normally think of as being infectious in any way. And so in terms of the microbes, I'm sure most of you will know this anyway, there's probably at least 10 million microbial genes if you compare that with the 21,000 protein coding genes in the human body, 100 trillion organisms, 10 times the number of cells in the body, more bacteria in your body than the number of humans that have ever lived or probably ever will live. So they're in there connected with us all the time. And they have a whole series of interactions with our body. We call them axes of interaction, microbiome host axes. So microbes make lots of chemicals that act as signaling agents that switch on and off processes in the human body, and change metabolism and change our phenotype. So microbiome is one of the biggest influences on the phenotype in its own right. And all of these chemicals are produced by combinations of bacteria, and they're signaling all the time to the body, and they're signaling through short-chain fatty acids, through bile acids, etc. And so in order to understand future disease risk, we have to understand that complex symbiotic biochemical interaction. And it really is complicated. So all of those different microbes, there's 2,000-3,000 recognizable species at any one time, but there's millions and millions of genes. All of those have got their own metabolite profiles, and all of those metabolites interact with humans. They're absorbed, metabolized, and they have pharmacological effects. So let's now talk, that's the background of the challenge. So let's now talk about the science and the enabling technologies, and what do you need to have a phenome center? Why would you have a phenome center? So a phenome center is basically a lot of analytical technology so that you can do vast numbers of analytical procedures that measure metabolites and metabolism, and in some cases proteins as well. So you can deal with large-scale epidemiology studies, you can deal with biobanking studies. You also have to have a capability of doing high throughput, basically metabolic information processing. You've got to be able to do something with those data. You tend to have multiple analytical modalities, and you have standardized protocols, right? So everything is uniform, and you can add to databases one after another. Every study you have increases your performance. And in an ideal situation, when you have a phenome center network, there's multiple phenome centers, they have identical protocols and technologies so they can share data. And also, you've got to have a repository for information, both the chemical information you measure, but also the biological. And in a big phenome center like ours, we're doing constant technology development. So we're improving things for the future, but trying to keep the basic protocols and the information recovery as standardized, so you're adding to data by developing new technologies, not subtracting from it. Philosophically, we've been using metabolites to measure metabolism for a long time. So this is from an old text written in 1507, where there's descriptions of urine in terms of smells, colors, and tastes. Yeah, people used to taste urine, and to make diagnoses, and therefore link those diagnoses into different disease patterns. So that is actually early type of pattern recognition. We call that AI now, right? Where we are actually connecting things that you've measured with a knowledge database and making interventions. Well, we don't need to taste urine anymore because we have spectrometers to do these, both NMR spectroscopy and mass spectrometry. So these are the major information containing analytical tools that we use. They're the major tools that chemists use to do structure elucidation on compounds. So there's lots of different combinations of experiments, lots of different types of separation procedure for separating molecules from biological fluids. There's lots of different sorts of mass spectrometer. And you can use these interactively to cover the huge amount of metabolic space that is in the human body. You have lots of different statistical tools. There are hundreds of different statistical tools that you can use, and these are being developed almost every day, new ones. So we've got to keep track on that. They fit into three basic classes. There's what we call chemometric modeling, where we're doing pattern recognition, but classification of samples, which is where you get your diagnostics from. So classify samples, you can say why you've classified it. That would be a diagnostic biomarker, and that can be used potentially in the clinic. And then there's something we call statistical spectroscopy, which is applying statistical tools to multiple spectroscopic data sets to extract latent information by comparing two or more different techniques that have been measured on the same sample. That helps you with both chemical structure elucidation, but also with pathway analysis And the final one, which is less used, but we are very interested in this, is what we call statistical thermodynamics, where we're looking at the actual entropy changes that occur when you disorder a system, such as getting a disease, which is a disordering state. So you need a lot of machines, right? So I just had a quick look in the lab that you're building behind here. That's just starting position. The lab in Australia cost so far probably about $50 million. So it's quite expensive. We have 11 NMR spectrometers. I can't remember quite how many mass spectrometers, about 30 or something like that. And there's a range of them for measuring a whole range of different properties of biological systems. And at this side here, we've got the extreme end, if you like, the very high technology NMR and mass spec, the expensive stuff. And at that end, we'll have much smaller machines. So one of the aims is to use all the complex technology to do biological discovery about the markers that you're going to need. And then you refine those techniques so you can measure it on a small machine that could be used in a hospital much, much cheaper. So when you're measuring 10, 20,000 variables, you don't need all of those for a diagnosis. What you often need is sort of 5 or 10. So what are the 5 or 10, right, that you need to be able to put on a smaller machine? So the way we operate at the moment is that we've got a huge number of different possibilities of platforms, but we've cut it back to a sort of, if you like, a skeleton group. But it still measures an enormous amount of information. So we measure lipoproteins, proteins, we measure lipids, we measure inflammatory markers, cytokines and things like that. We have amino acids and tryptophan pathway stuff. We've got quantitative targeted assays, and we do immunology screens as well. You put that all together, you've got an enormous amount of data. The important thing is that every sample that you take, that could be urine or blood, that goes through all of those different procedures. So each sample has an enormous amount of information about it. That's from an analytical point of view, but it also has a huge amount of metadata, right? So that's all the stuff about the patient or the individual. So there's all the stuff that doctors normally collect as part of their routine. They get the background, obviously male, female, age, and all the other stuff that you can collect. So it all has to be put together mathematically, and a phenome center is geared to running multiple studies at the same time. So I'm not quite sure how many we're running in our lab at the moment, but could easily be 20 or 25. So you get parallel studies, multiple analytical technologies, multiple data sets, and you've got all this metadata, genetic and clinical and physical, and it all has to come together into an informatics tool, a model, which you can then visualize. And ultimately, we want to be able to visualize it in a way that doctors can make decisions. So let's just very quickly go through some of the analytical technologies. I'm an editor of a number of different journals, and one of the things that drives me crazy is when I see that the opening sentence says metabolomics is an emerging tool or emerging technology. That's not true. It's actually been around for 40 or 50 years. This is a review we wrote in 1989, which makes me feel very old. And if you run through it, it's got all the things that people talk about in metabolomics and metabonomics. And it was there actually before people were doing proteomics, really. So the technology's been there and people have been making use of it for these sorts of studies, but they haven't necessarily put an omics on the end of it. And very early on, when we did a lot of toxicology work, we discovered that quite complex and distinctive patterns about metabolism are encoded in the spectroscopic data in themselves. So if you look at these are urinary NMR spectral patterns from these are controls, and these are different rats that have been treated with different toxins. You can see by eye that they're dramatically different. And what we've done over the years is refine the tools for recovering information. So there's a complex NMR spectrum of urine, which takes a couple of minutes to run in modern machines, right, with no sample pre-treatment. So it's ready for high throughput. And then what you're trying to do is get the information out of it. So there is basically NMR spectroscopic chemical information. There's biophysical information in there. There's biological information because the molecules in there are there because of biological processes. And there is clinically relevant data, which we call translatable biomarkers, all present in that spectrum. So that represents an informatic series of procedures to go from the spectrum which is pure chemistry, to the translatable biomarkers, which are clinical. And we spent the last 30 plus years developing that at Imperial and more recently in Australia. And once you've got the protocols harmonized, it means that you can set up inter-laboratory comparisons very easily. So NMR is particularly reproducible. So it particularly lends itself to this sort of harmonization. So you can get results in the UK, in New York, and in Western Australia on the same spectrometer. They'll be virtually identical. And that allows you to compare the biology of disease in different countries using an analytical technology. Now, you can do the same thing with mass spectrometry, but it's a lot more difficult to harmonize. And I'll say a little about that in a moment. So in our laboratory, the first stop is always the NMR spectrometers. We've got a whole range of them, and they're set up for different purposes, but we've got them parallelized so we can run very large numbers of samples a year. We can probably run 250,000 samples a year if we put all our NMR resource together. And that wouldn't be just one analytical procedure, probably 5 or 6 different NMR experiments for every single sample that we put through. So you can start to bring in put together enormous databases of biology very, very quickly. And because NMR is highly reproducible, you can compare it with data that's been collected in other laboratories as well. There's also the mass spec laboratory. We've got lots of parallelized mass spectrometry, so we can do large numbers of samples as well, simply because we've got a lot of spectrometers. We have very high-end equipment. This is our 800 megahertz system, which was put in last year. This is a very, very expensive top-end machine. It's good for a whole range of things, but chemical structure elucidation is what we mainly use it for. And we've used this to really good purpose in the last few months to identify completely new biomarkers of COVID-19 infection, which look as though they'll be relevant to a range of other viral diseases as well. And in parallel with that, we have the very advanced mass spectrometry. This is Fourier transform mass spectrometry. And this is ultra high resolution mass spec. This is a $4 million machine on its own. So we wouldn't call this routine, but when you're trying to do very high-end structural elucidation, having Fourier transform mass spectrometry is very powerful. So all these different technologies have to be put together in a particular way. So we can use them in the clinic for looking at populations. Population phenomics, so we're looking at large numbers of samples from epidemiology studies, but we can also use them in the clinic directly for trying to get new biomarkers of disease, and that's called clinical phenomics, and we're looking at new diagnostics and prognostics. We can also look at the patient journey. That's the journey through through the disease process, from going into hospital or even before his early stage hospital, then after an intervention, and then later, and through time, using these technologies to monitor the progress of disease and how different interventions change that progress of disease in terms of a phenotypic space. And that's very, very powerful. And I'll show you that in detail tomorrow for COVID-19. Okay, what about populations? Well, we started a lot of our population work at Imperial, and this continues now with our collaborations both with Imperial and in the United States. And one of the big projects we started on was something called Intermap, which was set up by this guy, Jeremiah Stamler, the father of preventive cardiology. He died last year, aged 102. It shows you epidemiologists live a long time, especially people like Jerry. He practices what he preaches, and he would never have salt in his diet, and he would exercise every day till he was about 101. So it's quite impressive. And one of the studies we looked at first was this Intermap study, which has got 4 different countries. It's got China, Japan, the UK, and America. And we just looked at metabolic phenotypes and how they mapped. And what you can see, this is just a hierarchical cluster analysis. What you can see is you can classify people straight away by their country. And so you have China down here, and you have Japan, which is similar-ish to China. And then they've got America and Britain, which sort of acts as 51st state in the middle of America. And it's quite interesting because if you superimpose sort of health on that, the healthiest people are in southern China. They've got a very good diet. And the most unhealthy are the people that live in the southern states of America. And so because where you live, it's your environment, has a huge impact on your longevity and the biomarkers, the biochemistry of your body. So you can ask the question, well, where's this genomics? These people are all different genomically. So what about Japanese Americans, right? So we had a few hundred Japanese Americans in this study. So they're Japanese genetically, but they live in Japan. Well, they map there, they're Americans. So once they live in America, they become Americans because they get all the American environment, and they get, with that, the American diseases as well. Okay. We use these data to create something called the metabolome-wide association study, which is not scientifically analogous to the genome-wide association study, where you're looking at combinations of genes and how that predicts future health problems or mortality. Well, here we're asking the question, can you look at a metabolic profile, urine or plasma, say, at a particular point, and then from that, try and predict whether somebody's likely to have a stroke or cancer in the future? And the answer is actually, to a certain extent, you can do that. And we are refining those technologies now. In this particular paper, we're actually looking at metabolic signatures associated not only with diet, but also blood pressure diversity. And of course, high blood pressure is a leading cause of stroke. Also, the obesity, adiposity is tremendously important. I showed you earlier about how obesity drives type 2 diabetes. So we can start to look at the metabolic signatures associated with obesity. And this is a whole series of different variables, which are very strongly statistically linked to how fat you are, basically. And all the red ones are related to the microbiome. So the microbiome actually has a very statistically important influence on how fat you become and how likely you are to get diabetes. So that's really critically important to think about that in terms of long-term healthcare research and planning. And also the metabolic variation or variance is much more closely related to obesity than the genetic variance is. So if you looked at this very, very big study looking at gene variation, we found that genes only explain about 1.4, 1.5% of BMI variation, whereas the microbial metabolites in urine alone explain 8%. So microbes have actually got a much more important influence than, pardon me, than the background genes. And you can put this together in terms of a network diagram. So you put all the metabolic data together in terms of the metabolites that are associated with obesity. All the ones in red are elevated, the blue ones go down. And you can start thinking about how that all fits together in a systems map. So all the different coloring is to do with the major pathways. So we can put an ontology on it. And we can also introduce the microbial parts, and so you can look at symbiotic biology at the same time. So we can look at the interaction between the microbial metabolites in a pathway sense that relates into the different obesity markers. We can also do studies on dietary interventions, right? So we're now into nutrition. Of course, there's a lot of interest here in Kaen University here. And this was an interesting one where we were looking at African Americans versus Africans. So African Americans obviously came over from Africa. So they have African genes, if you like, but the incidence of colorectal cancer in America, in African Americans, is about 40 or 50 times higher than it is in Africa, despite they've got the same basic gene subsets. And so the most likely thing is it's diet. So we did an interesting experiment a few years ago where we swapped over an African diet, which is pretty healthy, with an American diet, which definitely isn't. And you could watch the shift in the microbiomes before and after, and basically Africans became Americans and Americans became Africans just by a dietary intervention over a 2-week period. So you can really shift somebody's biochemistry, and associated with that, their disease risks, and you can express their disease risks, or you can calculate that from some of the precursor cell types that are related to cancer. And we biopsied all these people. So you find that over the dietary intervention not only changes the microbes to a healthier state, changes the metabolism to a healthier state, it actually changes the cytology to a healthier state as well. And you can put all of that together in a really scary looking diagram, which is the metabome networks, of how all of those things are interacting in the human body together. I'm not even going to begin to go through that with you, but we're now doing systems medicine on populations of people, monitoring interventions. That's what phenomics allows you to do. And this puts the whole of nutritional biochemistry on a new footing. That's not been possible to do before. You know, food scientists and nutritionists tend to do hand waving stuff, lots of red meat is bad for you, chicken is a bit better for you, fruit is good for you. Now we can put all the molecular basis behind this and look at it in terms of human systemic biochemistry. Well, in clinical metabonomics, we're looking at patient outcomes. So we go back to the patient journey and following it through. And there's a whole series of different areas that we develop. This was mainly done at Imperial, and we're doing it in a slightly different way in Australia at the moment, actually mainly because we've been dominated by COVID for the last 3 years. We put a huge amount of effort into COVID research, which, as I say, we'll talk about tomorrow. But almost every type of clinical journey, every type of disease can be looked at with this phenomic technology to produce a phenomic model of the disease. So here we have publications in all of these different areas, applications of phenomics in those, and that's not exhaustive. So I'll just give you one very quick example on liver transplantation and the need for liver transplantation. So if you get chronic and on top of acute liver failure, that's called decompensated liver cirrhosis. You will die from that, and basically unless you get a liver transplant. And in the UK, we have an opt-in system where you have to have a card in your pocket that says, if I get run over by a car, then transplant my liver or my kidneys or whatever. There's actually a shortage of livers in the UK for transplantation. So we were trying to think about how you could get a better model that would say, of all these patients, who needs the liver most urgently? So we took a training set with about 80 patients with decompensated liver failure, and then we had a validation set that was a bit bigger than that, and we had some controls. And we did the NMR and the mass spec on their blood plasma. And we built mathematical models that predicted outcomes. And not without going through the detail, what we could show is that whether you use NMR or mass spec, it was actually better than the current gold standard, which is the model for end-stage liver disease, which clinicians use routinely. But this was faster and cheaper, more sensitive and more specific if you use phenomics. So there's enormous possibilities there for future improved treatment and optimization of treatment, and in this particular case, selection of who's going to get the first liver transplant out of a limited number that's available. So that's just one example. Elaine will talk about other liver examples shortly. The other thing that's quite interesting is the NMR and the mass spec give totally and utterly different information. So these are all the different NMR model metabolites that are statistically significant. These are the ones that come out of mass spec. There's lysophosphatidylcholine, phosphatidylcholine, a range of different things. So you got two models that are almost as good in terms of sensitivity and specificity, but they're totally different parts of space. So which one are you going to use in the clinic? The answer is the one that is fastest and cheapest and easiest to use. Not necessarily the one that gives you most information, the one that gives you the right diagnosis in the shortest time with the least expenditure of effort. When you're looking at mechanism of disease, then you need all of it. Because if you just have the NMR data, just have the mass spec data, they wouldn't give you the complete story. You get completely different interpretations of what the disease was in the two technologies. Put the two technologies together, you have an understanding of what's going on. I'll just say a couple of things about mass spectrometry and imaging, because that's actually another important part of a phenome center. It's not just body fluids, we also do tissue imaging. And we developed this again at Imperial. This is using desorption, electrospace, solvation, ionization, imaging. And basically it allows us to take tissue slices and build mathematical models of the spatial distribution of the biochemistry and the differences between the different pathologies. So this is a biopsy of a gut tumor here, it's an adenocarcinoma. And what we're doing here is on the left-hand side is the histology, on the right-hand side is the mass spectrometric image. And we can take little sections of the tissue in terms of, and these are built up by rastering a beam of analytical beam across the surface of the tissue. And so we can build a mathematical model that helps us distinguish normal, abnormal, the the tumor and the normal tissue. And then we can pick out individual biomarkers and say what they are, because we can do all the structural elucidation. We can correlate the biomarkers and produce network maps. So this is digital imaging of tissues that produces not just give you a classification of the tumor, but will actually tell you about the detailed biochemistry. And it turns out that heterogeneity is also critically important. I know there's a lot of interest in liver tumor and liver pathology here. From a genetic point of view, when a tumor metastasizes and moves from one place to another, it has a sort of genetic evolution as it goes around the body. So there can be lots of different genetic subtypes, and even a single tumor can have multiple subtypes present within it. And so that's one of the big challenges, is that different tumor subtypes from a genetic point of view have got different levels of drug resistance. So a drug treatment might work on quite a lot of the cells, but it might not work on all of them, in which case you've created a selection pressure for a tumor that's resistant to the cancer therapy, and that's a bad thing. We were able to show a couple of years ago that you could use this imaging mass spectrometry approach to produce the same sort of mapping procedure to be able to separate out different subtypes of tumors. So the different genetic subsets have a metabolic fingerprint which you can pick out. And that makes this much, much more practical than just saying it's cancer, it's not cancer. You can say what sort of cancer it actually is. And we could do some fairly sophisticated stuff with that's a liver tumor in there with some, that's the liver tumor, that's the normal liver. And this is the mass spec image of that piece of tissue. That's the histopathological stain section, and this is the mass spec image. And this has just been imaged for molecular biomarkers of cancer. This bit is empty because that's the normal tissue. And then what we see here is all the different colors are different phenotypes of the cancer itself. They're actually different subtypes of the cancer. And this sort of imaging information is extremely difficult to get by genomics, partly because the size of a genomic sample is hundreds of times bigger than the bit that's analyzed by the mass spectrometer. So the image heterogeneity can be visualized with mass spectrometry at the micron range, whereas you're at the best really at the millimeter range with genomics. You can even see this rather spectacularly. This is serial sectioning through a piece of a liver and a liver tumor. The normal liver is in green, and that's just how a biomarker set varies in 3D. So we can start to see how these different pathways interact in complex ways. So this is hugely complex. I mean, that required that image has 725 million discrete pieces of metabolic information to create that image. That's quite extraordinary. But ultimately, we want to get out of this are models we can use in the clinic, where you've got a handheld supercomputer portal, all that data is coming through, and the clinician can access that just as well as you can access the patient's, bedside charts. I'm going to go through because I'm running out of time. I'll just say the last couple of things. Image guided surgery, that's a tremendously important development. It developed, I was one of the early developers of this, but the person who's really famous for it is Zoltan Takats, who I hired to Imperial, I don't know, sort of 7 or 8 years ago now. And we developed a lot of these tools here. This is the idea of using the products of diathermy. So the knife that cuts you, the electrothermy knife, generates smoke, and you put the smoke back into the mass spectrometer. And what happens is the mass spectrometer can measure the chemistry of the smoke that's created by the surgery about 3 times a second. It measures about 3,000 metabolites, about that sort of rate. It's quite extraordinary. So what we do is we basically create mathematical models that describe different types of tumor biochemistry, and the mathematical model can be displayed in real time to the surgeon. So it's cutting, so that's tumor, that's not tumor, etc. So you can start to use that information to clean the wound. So when you cut out, say, a lump of tumor, breast tumor, for instance, you can use the eye knife to actually make sure there is no tumor left. And that is in clinical trials in London for the last couple of years now, where we're seeing if we improve and optimize tumor surgery by using mass spectrometry. So it's in 3 different operating theatres in London. I'm not going to go through how the mass spectrometer works. That's a different lecture. And then, in fact, you can also do some amazing stuff. This is stuff that was Sultan Takat's group and a group from the Institute of Cancer Research linked to us, where they're actually using the information from the eye knife, not just for diagnosis and also for optimized resection of the tumor, but from discovering fundamental biochemistry about the tumor that relates to the process of tumorigenesis and also tumor growth. Again, I'm not going to go through all the different biochemical pathways here, but suffice it to say, we use this eye knife technology to find about enhanced arachidonic acid biochemistry, which relates to the mechanism of tumor growth. And we discovered that if you actually restricted the fat intake in the diet, you could use that to restrict tumor growth. Right, so that's extraordinary. Right? It's a dietary intervention that can help you reduce tumors. And here's all the background biochemistry. If you want to understand it, read the Cell paper. There's a lot of it in there. Okay, you can use this crazy stuff as well. You can use it for food analysis. So that's the previous Chancellor Vice Chancellor, the Exchequer in the UK, cutting into a piece of meat. So you can instantaneously diagnose food. You can diagnose it for contaminants, or whether it's decomposed, what type it is, whether it's Aberdeen Angus steak or whether it's Wagyu beef. That all has a signature, and it's basically instantaneous. And we were involved in a lot of the development of that, both at Imperial and also more recently in our labs. Finally, I just want to say something about networks and why it's important. We started doing harmonization and standardization quite a long time ago. We were the first group in the world to do this, publish this back in 2005. And what we really was making the case was, if everybody's got different analytical technology around the world, it's really difficult to compare the biomarkers of disease and use them in a meaningful clinical sense. Clinical chemistry and clinical diagnostics are absolutely standardized and regulated. So you need to be, if you're going to use metabolic phenotyping for clinical diagnosis and prognosis, it has to be standardized. And so we started to discuss all that. There have been many other papers on this since. But this is what you need for a phenome center: completely standardized protocols, which you can then harmonize with other groups. The other reason that you need this is that modern science is incredibly irreproducible. This is for a couple of years ago, but the estimate was, I think it's 2018, that every year in the United States, $28 billion of research money is wasted because of non-reproducible analytical chemistry. So that's completely hopeless. We can't do that in the future. There's not enough money in the world to do this. So science has got to get better and more reproducible. And so that's part of the mission of the Phenome Centre and the Phenome Centre network. Phenome Centre in London, we built the first clinical Phenome Centre in 2009, actually, and the first population national Phenome Centre in 2012. And we started to develop an international network in about 2013 or 14, and we formalized it back in 2016 at a big conference in Doha. And we brought in the Chinese as well. Chinese always like spectacular things. This is the launch of the Chinese Phenome Centre in Fudan University. A few years ago, you press a button, you watch them lift off the centre. It's all a bit show-busy. Anyway, what do? We're interested in all the biggest problems in the world. We work together to have harmonized approaches, international approaches to solving biggest problems in the world. So there are all the biggest problems in the world. There are some of the countries that have got phenome centers that are working with us. And 3 years ago, COVID and long COVID became the world's biggest problem. And so what we are trying to do, and I'll do this in detail tomorrow, is we're going to try to connect together population phenomics and clinical phenomics. So these are human population. People have got different biochemical backgrounds, different genetic backgrounds. When they get infected by something like COVID and they move into the disease, the outcome is dependent on their starting position in that metabolic space, on their background phenotype. People talk about susceptible groups, people who are obese, people who've got diabetes, people who are old and more likely to get severe COVID. And that reflects a mapping in a metabolic space that can be explored numerically. And so we combine that with the new spaces that people go into when they've got COVID, and long COVID is a partial recovery, but not complete recovery in the metabolic space. And in fact, many people we now know probably never completely recover from COVID. And those that appear to recover from COVID, they may start off in one space, but they end up in another. And that relates to what I started off with, saying change in the disease risk is calculable using phenomic properties and predictable for the future. And that's part of our major research program, which I will discuss with you tomorrow. So I've now finished, you'll be delighted to know. And I would just like to thank you for your kind attention, and also to thank the group here at Imperial and in the other universities that we work with. Thank you very much.
Thank you so much, Professor Jeremy Nicholson, for this inspiring talk. And I'm sure we've seen various use for the metabolomics and phenomics technologies. I think we have just some time for one very quick questions. Is there any questions from the audience? Could you just quickly mention, professors, on we see already various use of the phenomics and metabolomic technologies. In your opinions, what would be the next big things that we should look forward to?
The next big thing is, well, we're trying to do the next big thing, which is actually moving from the massive laboratory to the smaller laboratory, not necessarily in physical size, but in terms of use of the smaller equipment, because these are extremely expensive laboratories. You're talking about 50. To do a national phenome center, you really need $50 million, right? Well, they're not going to be everywhere. There's only going to be one or two in a country, but the possibilities for phenomics are enormous. So what you use is the big thing is that translation from doing big discovery type work, the small work, where you can have every university and every hospital can have a subset, and they're using the data and the models that have been constructed for whatever disease they're studying that have been done in the big laboratories. And that is a translational journey that we are doing now, but it's going to take 5-10 years before it becomes really widespread.
Yeah, I'm sure that we'll be looking forward to that translation into a more accessible approach for doing this phenomics research. So thank you so much, Professor Jeremy Nicholson, for this inspiring talk. And as Professor mentioned just now about another talk tomorrow, if you enjoyed this talk today, I would like to have more from Professor Jeremy Nicholson as well as from Professor Ellen Holmes. That's also another talk tomorrow morning on the title of Functional Genomics and Phenomics-Based Approach for Microbiome Host Interactions. If you have not registered to this talk but would like to attend, please feel free to contact the registration desk at the front during your lunch break. So that's another talk coming as well tomorrow morning. So for today,